Welcome, everybody. I just want to say thank you for attending our talk today. Our presentation will be from Jeffrey Townsend, who is a professor of biostatistics and ecology and evolutionary biology at Yale. So, Jeff, I'll turn it over to you.
Thank you for the invitation to speak today, and I hope I have something of interest to folks here. The title of my talk, as you can see, is The Durability of Immunity Following Infection by SARS-CoV-2. I'll explain more about the durability of immunity and why it's important in a moment, but I just want to mention that, of course, many of you realize it's probably important for a variety of reasons, but I just want to mention why I say SARS-CoV-2 rather than with COVID, which is that in this study, I'm actually looking at the virus and its relatives, and so that's why I'm sort of attributing more to the virus than to the disease. And so I think the title is fairly self-explanatory besides that, and I'll just take a moment to point out the people who contributed to this work at the beginning. It's work that I started just a couple months ago, and the primary person who executed the analyses was Haley Hassler, who's working as a research assistant in my group this summer especially, and then a couple other people in my group, Sayaka Mayura, Sudhir Kumar, are both collaborators of mine at Temple University who just executed a few analyses using different programs basically to make sure that it was robust to which methods we use, and turned out it was, which is great. Allison, many of you know, and then Alex Dornberg is a close collaborator of mine, works with me on many of the phylogenetic projects that I do. He's at the University of North Carolina, Charlotte. And I'm in the Department of Biostatistics at Yale School of Public Health. To get right into it, let me just start by mentioning that the durability of immunity upon natural infection has been called by many folks the greatest unknown factor of the COVID-19 epidemic. So on the left, you can see just a screenshot of a Nature video that Nature Publishing Group put out, that COVID-19, the big question 6 months on, and the biggest question that they said was, what is the status of immunity after someone gets infected with COVID-19? Another example is the online journal STAT, which published 7 months later what we know and don't know about COVID-19, the pressing questions that remain, and the number one pressing question they listed is, what is the durability of immunity to this virus after infection and after COVID-19 disease? There has been one major study, you could maybe count it as two major studies that has come out that had something to say about this and that got a lot of media attention, and that was a study of neutralizing antibodies to SARS-CoV-2, which showed that they dropped significantly in 2 to 3 months. It's still on medRxiv. As far as I can tell, it hasn't actually come out in a journal yet. It's been there for a while. I'm not quite sure what's holding up. In general, this was that was widely covered in the media. And the result you can see over here, this is from their medRxiv posting. These are just different antibodies to the N, the RBD, and S genes. And it's looking at IgM, IgA, and IgG optical densities of agglomeration of the antibodies. And these are the severity scores from red to purple of the disease of the individual. And the bottom line is that over time, you can see that all of these curves go up and then down again, indicating that there's a response to disease, increase in antibodies, and then a decrease later on in time. I would note that the error bars get pretty wide toward the end, which is where the decline arises. So the extent to which they've declined is actually not so clear, although it is clear they are starting to decline. And so whether it really has much to say about immunity is in question. And in the news, it was often mentioned as, we may need to worry. Coronavirus antibodies may fade fast, and it raises questions about vaccines. It sparks some fears for a little bit that immunity wanes quickly. On the other hand, two colleagues of mine, Akiko Iwasaki and Ruslan Metsitov at Yale, wrote an op-ed immediately after that sort of scare came out, pointing out that dropping antibody counts, even if they did drop dramatically, which it's not exactly clear they are at this time scale, they aren't a sign that our immune system is failing or an omen that we can't develop a viable vaccine. And they don't even really tell you that the person's not immune, since immunity is in part T-cell mediated, which you can't tell by measuring the antibody concentration. So that research was of interest and is important from immunological perspectives, but doesn't really tell you whether or not immunity is waning. And as I said, the error bars are pretty high. And so we can be a little trepidatious about interpreting that. And we really need a longer term follow-up before we really know what that means about immunity for individuals. Does that mean that we have to deem this question impossible to address? This epidemic is fairly recent. It was such a dramatically fast-moving epidemic that it is recent, even though it's spread across the entire globe. Is it so recent there aren't enough reinfections to actually estimate this? Well, there are news articles, one claiming that basically you can get it twice, and so there's no hope of immunity. Basically, another one claiming that it's very unlikely you'll ever get COVID-19 again. I think resolving this issue would be a really important thing to do. And we endeavored to do so using information that we all have, which is information about the evolutionary relationship of SARS-CoV-2 to other human-infecting coronaviruses. So here's a list of human-infecting coronaviruses, which, as I'll tell you in a moment, these are all the ones that have actually been studied for durability of immunity. It's quite a few of them. Fortunately, coronaviruses, there are quite a few pathogens of humans in coronaviruses, and there has been research on them, not as much as we would like, but there has been. And what you can see here, first on the upper side, the molecular divergences of SARS-CoV-1 to SARS-CoV-2, human virus HKU1, NL63, MERS, OC43, and 229E. And you can see there's quite a range of genetic divergence between SARS-CoV-2 and these viruses. I'll just comment to say that although it ranges down to as low as 19.9% divergence, these are actually easily alignable DNA sequences. So it's pretty easy to do analyses of the genetic divergence of these and actually align them, and then do phylogenetic analysis, which is what I'm going to talk about, what we did in order to actually ascertain the durability of immunity to SARS-CoV-2. In particular, you can also do time-based analyses, and you can see that the divergence from SARS-CoV-1 by our analysis is not so ancient. It's maybe a couple hundred years ago. And then other ones have deeper divergences. But even in the deeper divergences, we're still talking fairly recent divergences time-wise from the other viruses that are also infecting humans. That is reassuring in the sense that if it's fairly recent divergence, then that tells us that, in fact, these other viruses probably have something to tell us about the durability of immunity, because you can't change from one kind of entity to another very quickly. There's only one known mechanism of how these things interact with human immune system, essentially, like SARS-CoV-1, SARS-CoV-2 binds to the ACE2 receptor, et cetera, et cetera. So there's sort of a lot of molecular biology that's conserved between them. So we shouldn't assume that they're completely different and completely novel and turn our blinders to the fact that these are all evolutionary related viruses. So what can we do with that? Well, just to back that up a little bit, we do know something about the durability of immunity to SARS-CoV-2 because we know the evolutionary relationship. This is a small section of the phylogeny, just looking at SARS-CoV-2 and SARS-CoV-1. And what you can see is these two are related to each other. And if we look at the different genes in the genome, there's the S gene, the spike gene everyone talks about, M, N, E, and then a number of unnamed, although we do know something about their function, open reading frames that are in the SARS-CoV-2 genome. And if we look at all of those and look at what their phylogenetic informativeness is, this is how much information they contain about how related they are to other viruses at different time depths, you can see that they're actually pretty nicely informative about the time depths we're interested in. We're interested in the divergence of SARS-CoV-1 and SARS-CoV-2. That's right here, close to the upper peak of the informativeness. And then as we go back, we still have pretty high informativeness for the other divergences to the other ones. So there's reason to believe that phylogenetically we can get information from these different viruses about each other. So the point is that these genes of the coronaviruses evolve at rates appropriate for phylogenetic analysis. So if we do that phylogenetic analysis, we also have benefit-dense taxon sampling. There's quite a few SARS-CoV isolates sequenced now, plus other isolates taken from a variety of bats, ducks, shrews, many other organisms, as well as the other coronaviruses that have infected humans. We also have sequences of those. And so there's quite a lot of sequence that we can do to analyze to get a phylogenetic analysis of the nucleotide sequences of, and in particular for all the analyses I'm going to talk about, we use the S, M, and ORF1b genes, because they're so essential to these viruses that they're present in all of them. And so we didn't have any problems of missing genes if we just use those three. Plus, the three of them gave very high support for any internode deep in the phylogeny. Only the very tips were unresolved, and those usually were very closely related to viruses like two different SARS-CoV-2 isolates, so it wasn't important to resolve the phylogenetic relationship to them. All these blue dots indicate 100% bootstrap support for their phylogenetic relationship. Okay, break down to the species we're interested in. We did the phylogeny for a much bigger phylogeny because it gives us more power to actually infer the phylogeny correctly, et cetera. But just so we can focus on it, I've pruned this just to the SARS-CoV-1, SARS-CoV-2, MERS, OC43, HKU1, NL63, and 229E coronaviruses. And this is their phylogenetic relationship to each other. All of these dots indicate, again, 100% bootstrap support. So we really know the topology of the relationship of these. And the branch lengths here indicate their molecular evolutionary divergence. And so what we did was we took this molecular evolutionary divergence tree and did a number of analyses with it, all of which ended up showing the same phenomenon, which I'm going to go to now. And I can talk about the robustness to lots and lots of parameters because we've done all of the robustness analyses and they come out very nicely afterwards. But just imagine for now, so you can understand the basic analysis, what we do is we take this tree and we do what's called ancestral and descendant states analysis. That's an analysis where we look at what the ancestors of these different viruses probably were like based on the amount of divergence from each other, and then we can even project forward to things that we don't have for a particular property. So if we look at durability of immunity, as I mentioned, except for NL63 and SARS-CoV-2, all of these species have previously been studied in various contexts for durability of immunity. So what we're able to do is take those measurements about the durability of immunity for all those other species and then infer what the ancestors' durability of immunity was, and then project that forward to the species we don't know. So just to narrate that a little more, here are the 4 viral species with their durability of immunity in weeks on this axis from 30 to 86, which is the full range of the durability of immunity across these species. What you can see is these HCOV, these human coronaviruses, the circulating human coronaviruses have fairly low durability of immunity, ranging between 30 and 44 weeks. In contrast, SARS-CoV-1 and MERS have a higher durability of immunity, between 72 and 86 weeks. And so there's sort of 2 groups here. And what we can essentially do with the ancestral state analysis is take those and infer what the ancestors must have looked like under a model of trait evolution. We use the Brownian motion model of trait evolution, which is the most common and accepted model to use for continuous state evolution. We have tried other models, result comes out the same. And so what you find is that we can infer what these ancestral viruses' durability of immunity was. And then, as I said on the last slide, we can then project forward for what the durability of immunity of SARS-CoV-2 is based on its ancestry with SARS-CoV-1 and with all of the other species going back to these internodes back here. And that provides us with an evolutionarily justified estimate of what the durability of immunity will be when we do a longitudinal study looking at individuals. And it's a far better way than just taking an average, which I'll show you in a little bit. So that's the durability of immunity of SARS-CoV-2 estimated via ancestral and descendant state analysis. It's 78 weeks or about 1.5 years. There's been a lot of talk that we just don't know this because we haven't done the empirical study we need to do. But actually, that's just ignoring the fact that we understand that evolution happens. And in fact, we have ways of using evolutionary biology to make inferences about how species are related to each other and what their traits evolve like over time. So this model basically is a model which assesses how does this trait evolve over time, how much does it diverge over time, and projects forward what the most likely durability of immunity is. Of course, it does have error bars. These error bars I show you below are robust to a vast number of things we've analyzed it, and you get almost exactly the same plot and the same error bars, depending on the phylogenetic uncertainty of the branch length, the topology of the phylogenetics, ancestral and descendant state estimation uncertainty, as well as we took kind of there was recombination blocks in the sequence. We analyzed it with just the non-recombining blocks. We get the same result. We also, as I mentioned, analyzed it with an Ornstein-Uhlenbeck, which is a different model, and we get the same result in terms of estimating the duration of immunity. Tremendous robustness to everything we could possibly throw at this, which is very reassuring. These are fairly wide intervals here, but I'm about to show you in the next analysis that I think these are probably too wide. There is a phenomenon in phylogenetics that of course, there aren't that many species that we have. We only have like 5, and we're projecting the 6th species, which is SARS-CoV-2 or NL63. And when you have very few species, then it's very hard to constrain the variance of the Brownian motion process, so you end up with wide intervals. That actually doesn't mean you have low predictability, which we were able to show by doing a counterfactual subsampling and reanalysis of viral taxa. So we take the viral taxa we know and say, okay, suppose we didn't know that one of them, or we didn't know 3. If we took what we did know, would we be able to predict what the known durability of the viral taxa is? In other words, we predict something we actually know and say, how different do we get when we make that prediction? And what you see here is that here we're looking at if you just take the average of a set of taxa, or if you take this tree-based approach we're talking about. And what you can see is in either case, you actually have a rapidly declining projected mean error as you look more and more taxa and use more and more taxa to estimate the durability of immunity. And in fact, we can go up to 4 here because this is, again, predicting if we take 4 of them and predict the 5th one that we know, what's the error in that prediction? You can see it varies quite a bit, but the expected error is under 10 weeks. In fact, it's about 8 weeks. Now, in fact, we have 5 and we're predicting the 6th. So if you just take an exponential regression, take this as a prediction with an exponential regression, what you find is that we're predicting about an error of just 2.5 weeks. So we're basically, empirically, if you analyze what our process does, without any assumption about the Brownian motion, just what does our process that we compute through here give us in terms of an error of the known things that we get, and then project that forward, we only have an error of about 2.5 weeks on 78 weeks of durability of immunity, which is quite astonishing, actually, and I'm very happy about that. So I've sort of brought you through all of this analysis, and I hope that I made it clear and that you followed it, and that if you have any questions, you'll be able to ask them. But I guess I'll go through my conclusions and caveats first. The conclusions that I have are that this ancestral and descendant states estimate of the timing of the waning of immunity can facilitate all of the quantitative analyses of policymaking with regard to individuals who recovered from COVID-19 who may be viewed as immune to reinfection. So an enormous number of the epidemiological models, at least if they have any long-term component to them that we run, need to have some idea of what the duration of immunity is. And if you don't have a quantitative estimate for that, it really leads to a lot of sensitivity analyses that are really problematic to presenting a clearer scientific message going forward. So I'm hopeful that this analysis and the result that we get can help people to focus on what the most likely durability of immunity is, so that we don't have too many different analyses to really make an interpretive conclusion or make a decision. The durability of immunity has implications for a lot of different practical things about what we do in response to this epidemic. It implies many things for the deployment of recovered healthcare workers, for travel restrictions, for decisions regarding how students obtain their education, for prospective vaccination protocols, for clinical trials, as well as for the opening and closing of economic sectors in response to predictive models of the epidemic. Our estimate strongly argues against the claim that a long-standing resolution of the epidemic could arise due to herd immunity from natural infection. This is somewhat simplified, but basically you can just sort of say such a strategy jeopardizes millions of lives, entailing high rates of reinfection, morbidity, and death every 1.5 years. If you only have about 1.5 years on the basis of a natural infection before you can get infected again, that's not enough of a timescale that herd immunity provides really much of a solution. By the time you sweep through the population once and get herd immunity, you're ready again for another epidemic. So we need to be wary of the idea that herd immunity is going to be a solution to this problem. It does provide some guidance as to the likely timescale conferred by a typical vaccine. Of course, vaccines can confer different levels of immunity than natural infection. And what we've estimated is the immunity on natural infection, not the vaccine immunity. But typically, a vaccine gives you similar amounts of immunity to what the natural infection does, although there are definite exceptions like the HPV vaccine you can bring up. This approach has general applicability, so it's not just true for COVID-19. We could probably do this for other novel pathogens, so I hope that we're able to do it at a faster pace because we don't have to develop the pipeline, basically, in future epidemics, because provided they're embedded in a clade that contains 3 or more previously studied human pathogens, we can do the same inference on just about any pathogen. It does depend a little bit on how diverged they are. As I introduced in the beginning, it's kind of important that they be fairly closely related, or else obviously there's less information about one pathogen by looking at the other that have evolved. And the other conclusion that I want to bring is just the point that I think is often overlooked, which is that evolutionary biology as a study, we often think about it as a historical thing where, we're looking back and trying to understand what's going on in the past. But understanding the evolution of things actually gives us predictive power, and it's something that is underappreciated, I think, among the scientific community. So I hope everyone understands that it is evolutionary biology that gives us this answer, and it's a very important study for prediction of things that are ongoing now. There are a couple caveats I just want to mention. This work is ongoing. So in particular, the results that I gave you today are results based on what I would call a quick and superficial analysis of the immunological data for those coronaviruses that we estimated the durability of immunity. We're now revising this with an immunologist collaborator who knows more about immunology. I don't think it's going to make any dramatic changes in our prediction, but it definitely will very likely have some slight quantitative changes. Those quick interpretations are there, and so just a caveat that this is not a final result. It's also not gone through peer review completely yet, so hang on to your horses, but hopefully we'll have a full result soon. Our estimate and uncertainty should be understood as a prediction of the average durability. It's not a universal durability. Not everyone is going to have that durability, obviously. The durabilities that we put in from the studies that we looked at are average durabilities, and so what we're inferring about SARS-CoV-2 is also the average durability, not any individual durability. Also, one other important caveat is that biases that are peculiar to this disease should be kept in mind. So SARS-CoV-2 in particular has this surprising degree of asymptomatic infection. And it's also clear from some other studies that asymptomatic infection can induce a weaker immune response than symptomatic infection. For that reason, asymptomatic individuals may have a lower durability of immunity than is captured in our analysis. And because the other species don't have that asymptomatic phase, I would definitely, if I were doing a quantitative analysis like an epidemiological model and using this data, I would acknowledge that it may be a lower durability of immunity for asymptomatic individuals, although we don't know that either, but it's just a point that that's something should be kept in mind carefully. Also, because SARS-CoV-2 is a novel virus, the human immune system, we need to keep in mind that reinfections may not exhibit the same severity as first infections. It may not be as bad a disease, but we also really don't know that. That's just a possibility. Those are my caveats, and I'd be very delighted to entertain any questions anyone has regarding this research.
Thank you very much, Jeff. Excellent talk. I'm sure lots of questions I have, but I'm going to let others ask first. Thanks.
Hi, Jeffrey. This is Chris Barrett. Great talk. Have you had a chance to look at different locations in the world with enough detail to track the impact of response on the genetic history of these things? That is to say, do the local responses seem to affect the virus itself in terms of fast-moving evolutionary history that you're talking about? Is there any evidence for that you're...
Oh, I see what you're asking. Yeah, so in this particular study, we're looking at deeper evolutionary phenomena than it's true we had many like SARS-CoV-2 viruses in the phylogeny, but from the perspective that we're looking at from this in terms of the depth, those are all very, very tip lineages. They're all so closely related to each other that it really makes no difference to our analysis, but it doesn't hurt our analysis, so we kept them in there. So in that big tree, you saw lots of very closely related SARS-CoV-2 genomes. So for this paper, for what I've described just now, we haven't exactly looked at anything about the later evolution of the coronavirus in response to human interventions, et cetera. And I have not looked at that. So I can't really properly answer that question. I always like to give some answers. The one answer I'll give you is that in another study that we're doing right now, we are looking at not quite the timescale you mentioned, which is like how does response affect the evolution of the coronavirus itself, but we are looking at the zoonotic period. So if you take all of those tip lineages and look back towards what their common ancestor is, all the ones that infected human, and then look at the ancestral lineages that infected, say, bats or pangolins or shrews or whatever, and then find the most recent species there, we have spent some time looking at that intervening evolution between what was the pre-zoonotic pathogen to the zoonotic pathogen. People often speculate, like, did it evolve so that it could infect humans? And I have a whole talk I can give on this, but the short and sweet of it is that it doesn't appear that it's really evolving a lot in order to infect humans. It looks very much like it just was there ready to infect humans and moved in. And the reason we can say that is we just looked at whether there was natural selection using genetic techniques on the changes that occurred between the ancestor in the wild host to the human host. And you really see very little evidence of that. And that's repeatedly. So even in the original SARS and in SARS-CoV-2, both, we've done that same analysis, and we really see very little evidence for natural selection for divergent function. And again, that has to do with the fact that, in fact, we're not that far evolved from the bats and the shrews, right? I mean, we're all mammals. We all have ACE2 receptors. They all have the same structure, even though they have some genetic differences. Most of the functional parts that are the parts that it binds onto are pretty much structured the same. And that's why we can use mice as a model to try to understand these things, right? All of this says that we're not that different from all of our evolutionary friends in the world today. And so for that reason, there's a lot of power in understanding, what's going on for us, understanding with them. And so consequently, when the virus is circulating in bats, it may just be a matter of actually getting a transmission, which you don't usually get up close and personal with that, in order for an infection to happen. And then it has to spread from that individual to someone else and reach a higher number. As we all know, there's a lot of stochasticity at the low numbers. So I think a lot of it is just a random transmission. And it just means that we need to be very careful with the kinds of ways we interact with wildlife and respect that boundary. So I know I didn't answer your question, but I hope I gave
you answered many aspects of the question. Thanks. Appreciate it.
This is Ron Kenyon. Great talk. I'm just trying to draw some additional conclusions from what you were saying. It sounds like if you have a immunity that is only going to last a year and a half, and as you said, the herd immunity is not going to solve the problem because it's going to start cycling back through the population. Yeah. Does that indicate to you that we would basically just have to have a vaccine every year, sort of like what we do for the flu or something, to get it under control?
Yeah, I'm kind of two opinions about that. I think that we shouldn't count on not needing a yearly or biannual vaccine because of this prediction of 1.5 years. On the other hand, I do think that I don't know that much about this area, but I have looked at it a little bit, that the biology of the coronavirus infection is not all that complicated. And people have said this, we haven't generated a vaccine for a coronavirus before, mostly because people just sort of said, oh, it's common cold, we don't need to worry about that. Unfortunately, I think that that's a mistaken impression. I hope that in the future, we are more liberal in our grant panels to fund studies on mildly morbid causing disease, because those same species may someday turn out to be the important ones for really, really devastating disease. That's an aside point. Your question was with regard.
It was with respect to having to have a recurring vaccine, basically.
Right. So I think that we have to be cautious, and we can if we want to be conservative, I basically think we should assume it's about 1.5 years, and we may need a revaccination. So that's one area we might be in. On the other hand, I also think, just based on the biology, it may be that a vaccine, especially with the 126 or so candidates that we have, someone may be designing a vaccine that evokes an immune response that's very different from what you get from a typical natural infection. If you could get an antibody that digs in and gets onto that M gene, which is not usually accessible, instead of that RBS, which actually evolves very quickly, the spike protein, maybe that could give you a very highly lasting immunity to the SARS coronavirus. So I haven't reviewed all the vaccines to figure out what each of them are doing, but there are a lot of different candidates out there, which is really exciting. And I hope that we manage to keep funding all of them through to the point where they get completed, right? Because some of them could give us lifelong immunity, whereas others could just give us 1.5 years, is what I would say. And we don't really know until we actually try them all. I don't know if we're going to get to all of them. Obviously, some of them are going to have some complexities and some side effects that are problematic. But I do think that there's some potential that our vaccine may be longer lasting than 1.5 years. But I think we should count on our natural infection to not give us more immunity than 1.5 years. There's really no justification for believing that it would last a lot longer than that.
Okay, thank you.
Hi, Jeff. This is Andrew Warren, and I apologize if you covered this. I came in sort of late from another meeting, but I was curious about, and this may be clarified in your work with an immunologist, but I was curious if, in terms of reinfection states, like how you draw the line or even quantify it. Is it from sort of titer measurements for viral load in the organisms in question for like MERS and the things that have been more well studied? And I was just curious about that association, making the binding from the literature to some sort of reinfection state.
That's a very good question, and that's why I put it as the first caveat here. Maybe I should have been clearer that I also am not exactly sure. Basically, we looked at the papers and we tried to understand what the authors of the paper said. It said about immunity, and that's what we put in. But I'm now working immunologist, and hopefully I can come up with a better justification than that for now. But we just looked at the paper, and the papers typically say something about on the order of something is about the immunity that we expect given this analysis. Some of them were based on longitudinal studies that involved looking at how many people were reinfected. Others had more antibody data. So the different studies are a little different. They do have overlap, so that's good because there's a way to make them all comparable for our analysis. But I think it really requires someone with more expertise in the immunology than I have to make that call. But again, as I say in that caveat, it's hard for me to believe it's going to be very different from what we estimated, just because that would require whoever published that paper to be sort of very wrong in what they said. So I think it's going to be right, but I don't think it's going to be exactly the numbers I'm giving you here. I mean, the end result could be the same, but I think certainly the results for the individual ones will change, at least in a couple of instances, because we're reviewing that now, that an immunologist is required to make that kind of decision. I can't speak to it very well.
Right. And it seems I was sort of a follow-up comment question was that I've been surprised by there's sort of a bicameral organization between these two different immunological responses, more of a long-term immunological response and a short-term. And I was also wondering if the assay types in all of these different studies, as we understand more, do you feel like they will be robust in terms of the type of immunological response, or whether viral load is just the overriding sort of factor, or whether you would need to be able to measure T-cell activity and antibody levels and so on?
I think, again, I definitely would refer this to more of an immunologist than I am for a really good answer. But the little bit that I can just say is that I think that the studies that are out there have at least antibody data going out for a long period of time. And so that when you get that longer period decline, and then usually there's some data on reinfection that can be sort of aligned with that, then you can come up with these, they call them, what's the technical term? Again, this is not my field, but I have reviewed this a couple times. Yeah, I don't remember the technical term, but it basically is the thresholds of, or biomarkers of immunity. Basically, anytime you have a disease, what you're looking for are antibody concentrations that correspond with immunity. And you have to do the right studies to connect the correspondence with the immunity. And you can't just look at antibody levels per se without any understanding of empirical immunity to understand what a threshold for immunity is. So that may be kind of what you're getting at. And my point would just be that, yeah, in the ideal case, what we'd have going into this was a ton of really good trials which looked at longitudinal possibilities of reinfection and monitored antibody levels during that. That does not exist for every one of these viruses, but there are some at least comparable immunoglobulin IgG antibody levels, which are the ones, maybe I'll just go back in my talk to here. One of the other caveats about this result is that these IgG antibodies, for instance, you can see they're only just barely declining, and these are longer term antibodies, et cetera. So there's a lot that needs to go into this. Again, I'm not really qualified to do much about the immunity. All I'm qualified to do is sort of take the 10,000 foot look and say, if I believe these numbers that are given in the literature, this is what the projection would be. I did enough looking at this to just say, okay, I think these papers are kind of basing things on the same general principles. So I didn't see that, one was a completely different kind of analysis giving a different result. So I feel some confidence that these papers can be compared. The degree to which you can really ascertain the immunity from antibody levels as opposed to a real reinfection study, which is what you really want, I'm not sure. But the one confidence that I have is, again, just like I mentioned, these diseases are not completely independent things where the amount doesn't relate between diseases. These diseases are fairly closely related. The viruses are fairly closely related. How they operate with the human immune system is fairly closely related. So I feel a little bit of okayness in general with saying, okay, well, in this study, if we don't have much reinfection data, at least we can look at the IgG values and the IgG values in the other study and say maybe it's comparable in some way. I don't think that's unreasonable because they're not that evolutionary diverged. So it would require that be a thing that specifically evolved a difference. And these are pretty constrained things, right? They have to interact with the human immune system, which is really trying hard to kill them. And there's not a lot of pathways that they can be successful in that enterprise. And so they're really conserved. It's one of the things that's kind of surprising because we always learn about influenza, which was mentioned earlier in the call, which evolves very, very, very quickly. You also learn about HIV, which evolves very, very quickly. But a lot of viruses really do not evolve quickly. They have a system that works for them, and they're conserved, and the proteins do not change in conformation. They do not have selected sites. They just are sticking with this plan that does work for them, and there's not another way that they can do it. So it depends on the virus, but these ones are pretty conserved, which is why you might have heard some of the vaccine people sort of enthusiastic about the prospects for vaccines, because the thing has one way of working and it doesn't really work lots of different ways, and it doesn't evolve very quickly compared to HIV or influenza, for instance.
Right. Makes perfect sense. Thanks so much.
Jeff, this is Madhav here. I have a question. Two parts related. One is technical, and it actually is my naivety, so excuse me if it's trivial. You try to make an inference for going up the tree and then also looking at the states, right? So there is an assumption and purely as a technical question, that if something below the ancestors have the same level of duration in some form, or how do we compute this? Because you're trying to then make inference down on the other side of the tree, right? That's from a machine learning standpoint, that was a question I was trying to tease out. So that was one. And related to it, if we start seeing the drifts that happen in this virus, like the influenza virus, I do not know how much the drifts are. Do you think that the drift would be such that the durability will maintain in some sense, or this adds another twist to the whole thing? Maybe they drift enough in a short period of time. You of course are looking at a set. Yeah. Sorry if it's too stupid, please.
No, those are great questions. I was sort of wrestling with how much I wanted to go into the Brownian motion model, which is the answer to your first question in this talk, because it gets very technical and phylogenetic. But there's actually a pretty quick way to just verbally describe it, so I'm happy to do that. The Brownian motion model is a pretty simple model of trait evolution, which is what we use to figure out what was that ancestor compared to the descendants. Under the Brownian motion model, what you're assuming is that the expectation for what happens to the trait is that it basically just stays the same. And you may have noticed it, that's exactly what you see here, is that when we go back to the ancestor, like this is the ancestor that we're estimating, and then we look at NL63, which is one of the two things that we estimated, or SARS-CoV-2, it's just a straight line, right? So that just tells you that the expectation under this model of the evolution of this is that it just stays the same. However, it acknowledges that there's a drift process, which is related to the second question you asked, where they go to different levels over time. And that drift process is a variance that increases over time. And so that's how you explain the differences that you see among these, is that, they expect to stay the same, but there's just a variance over time of how much they stay on that expectation or not. It's essentially, if you were to come up with a de novo sort of a hypothesis of how things evolve, where you said, well, basically I want to be somewhat conservative and not say that things evolve like crazy, you would use kind of a normal distribution and just say, well, it evolves according to normal. So that's basically what that model is. It's a very straightforward model. It's actually, and this is maybe I just assert from my experience that it's very hard to write a model that would turn out very different for continuous traits. No matter what you do, you end up coming back approximately to this Brownian motion model. So there's really not much of another way to do it. There is another model called the Ornstein-Uhlenbeck model, which I also used. And that model, unlike that one, says that in addition to that drift process and an expectation that things stay the same, there's also a selective process that is pressuring all of the pathogens toward one unspecified value. It's a parameter in the analysis. And so that would be more reasonable if you thought, this duration of immunity is what really works for these things, and maybe they're varying because of various other stochastic things, but there is some selection for them to head towards this one immunity. And somewhat to my surprise, under the Ornstein-Uhlenbeck model, it comes out almost exactly the same. And I think the reason is that it's a little bit different. They tend a little bit more toward the middle, but it's almost trivial difference. And the reason that they come out almost the same is that, as you can see, there's quite a lot of divergence here. And so that means that there can't be strong selection under an Ornstein-Uhlenbeck model either. And the variance of the Brownian motion model is actually fairly high as well. So essentially, most of this is just a drift process on an expectation. That's getting very technical about how we do it. These are very well-established techniques in phylogenetics and evolutionary biology. So I don't think anyone should be concerned about their applicability to the problem or their technical accuracy as a method for estimating the trait values for species. I think it's worthwhile to think carefully about, can you really think about this as a trait? Is it a trait of the species of virus? How much durability of immunity does it have? I think that's a valid conversation to have, but I'm quite confident that I would argue that it is a trait and that we can do that. You had another question, Madhav. You talked about the drift process. Compared to influenza, I saw one estimate, so don't quote me, not my work, but I saw one estimate that said that these coronaviruses drift at about 10 times lower rate than influenza. That's a significant difference, an order of magnitude difference in how fast the sites generally evolve. And I think that even encompasses this RBS domain, the part that binds the spike protein at the very tip, that evolves fairly quickly. So even that part is not evolving as quickly as influenza. So that's one of the, I have some feeling that it may be possible to vaccine this thing a little better than influenza, because it's not really an issue of that stuff evolving too quickly. It does evolve at a higher pace than the rest of the genome, which is why I think if we could target immunity towards some other region other than that RBS region on the spike protein, that would be really, really great.
Thank you very much.
So I can understand this method. Is it also assuming that the environment stays constant over this time? So in this case, the environment is the immune system of the organism that's being infected, for example.
Yes, yeah, it's assuming that the environment is completely the same. That's absolutely right.
Okay, so are there models that take into account, so because I think of immunology as like a two-person game, right? The immune system is also evolving and the pathogen is evolving. So is there a way to take that into account?
Maybe there is. I haven't thought about that, except to say that I have thought about whether I worry about that, and I don't worry about that. Okay. And the reason I don't worry about that is because the immune system evolves in a person. Like there's an evolutionary process in our immune system in ourselves, right? And there's a second evolutionary process, which is how, as humans, our immune system evolves. Right. And my feeling is that although the immune system evolves very quickly within ourselves, how our immune system evolves as humans does not evolve very fast. And on the time scale of the divergence that we're talking about here, as an evolutionary biologist, I don't think there's any real evolution of our immune system since 784 BC. I mean, that's,
Okay.
Not really.
Okay, thanks.
Hi, this is Anil. So really interesting talk. One question, which is, so like in your last slide, you had this bullet that reinfection might have lower severity. Does that also translate to the transmissibility? So like the contagiousness of the person who gets infected?
It may.
And is that something that is also tied to phylogenetics in some way, like how it transmits?
Yeah, absolutely. I think so. So yeah, and to answer your first question, it may well be tied to that. So in other words, it may transmit less the second time this goes around because individuals get less infected, just like I mentioned with asymptomatic. Maybe there'll be even a higher asymptomicity. I don't know. It's a little hard to make these predictions about whether things get worse or better because it depends on, there's a whole field that I have not dived into. You guys may remember Paul Ewald making statements about this some time ago, and then there's been a lot of people who've worked on it further, but it's actually pretty hard to tease out what what the evolution of virulence, so to speak, really is. You can analyze a particular system and come up with predictions, but it's very hard to make generalizations that are very, very robust about what direction virulence should evolve. And so I'll just caveat it by that. But what I will say is that this analysis, we were interested in durability of immunity. So that's what we put on that tree, and that's what we inferred for the other organisms. But any quantitative trait that you believe is like a heritable kind of trait, is answering your question more directly, you can on that tree and make a prediction for what the other pathogens are going to have where you haven't measured that trait. So it doesn't have to be durability of immunity. It could be transmissibility. It could be any of these quantitative traits we're interested in. So this approach is not just a durability of immunity approach. It's an approach we could use when new pathogens come in for lots of different things when we have like no data whatsoever on the pathogen. So it really does have, this is relevant to this particular GPCE effort, which is to try to address new pathogens when they come in. We should have this pipeline sort of set up and ready to run when a new pathogen arises so that we can try to get information about it before we even know about it. You know what I mean? I'm very enthusiastic about that. And I'm very enthusiastic about using it for other traits as well, where we sort of have done previous work on other pathogens and we have a new pathogen where we'd really like to know these things, but it takes time, obviously, to do all the studies to do it. To access what we have and come up with an estimate. Now, of course, our estimate is never going to be better than an empirical study that actually assesses that particular thing. But I'm not a believer in the idea that we should sort of put our blinders on and assume we know nothing just because we don't have an empirical study. Empiricism has its limits, and understanding that we do know something about things is very important.
Is virulence a trait? So can you make a tree like of the type you made for virulence as well?
Yeah, absolutely. You could do it on virulence. Let me give you a caution though. Durability of immunity, we got these results and they were very robust. Another trait that we put on there might have very different robustness. It might evolve much quicker, in which case there might be no predictability. So what I'm saying is we should do that kind of analysis, but you can't count on getting a result. It depends on whether it actually is slow to evolve, right? If it's slow to evolve, then we'll see that in our estimates and we'll see, we can estimate that thing. If it's evolving quickly, then we'll see the numbers have gone all over the place. The variance is going to be very high on that, very, very high on the Brownian motion or Uhlenbeck model. And essentially we're not going to be able to have much predictability on that trait at all. So we can put anything on there. Just be aware that it doesn't necessarily give us a result that's going to be useful for anything that we put on there. It depends on whether it's evolutionarily conserved among the pathogens we're looking at.
So Jeff, I think you pretty much outlined a program right now that takes important epidemiological traits that we are interested in from a modeling perspective. Anil had one question, durability is another question. There are many other facts, right? How quickly do you fall sick and become infectious? So they're all traits of a virus. And build a program out, which is computational in nature, and looks at zoonotic set, which Kathy, I think, is online here. She gave a talk last time. So you're a perfect program or a project for this expeditions proposal, because once you have this, you're in much better shape, and especially doing the computational studies. So I completely agree that we don't need to do deep tests to begin to do this. The computational experiments still provide a lot of value. But in doing that, one question that arose is, do you see a trade-off eventually in these viruses between the durability? People have talked about how the virus might become little less aggressive in its ability to make people very sick, but easy to jump from one person to another. It would seem that these traits the virus is trying to keep up have, and I do not know what the model is, but it cannot simultaneously optimize each of it, because if that would have, it seems to be tough for a virus to do. We have not seen too many of them. Is there a theory that tries to understand, say, 3 or 4 of them and tries to understand what the overall envelope of its effectiveness is? So if it's become very good in one, it might become weaker on the other side. And
you mean in one viral species compared to another?
One trait, right. So if this is SARS-CoV-2 becomes very good at infecting more people, it becomes more easy to infect. But maybe it's how sick you become, because that will have to necessarily weaken. Is almost like a limit law which says that you cannot simultaneously optimize all these features for these viruses.
I'm going to heap praise upon my evolutionary biology colleagues who have developed all the technology to do this already. Yes. So this program, we use phylopars to do what we did here. What we're doing right here is kind of this thing that these guys do all the time. It's just saying, okay, we have a trait, how does it change? But they've also mapped multiple traits and looked at the covariance of those traits and how they covary across the phylogeny. And the program, it's funny, the title of the paper for the phylopars program is all about those covariances, right? We didn't use those covariances at all in what we did because we're just looking at one trait. The level of sophistication of the analyses is quite high. It would be totally feasible to do those same analyses essentially with almost exactly this pipeline. It's just a matter of putting in more than one trait into the input.
That's very cool. Thank you very much. Yeah.
So we have one question from the chat. Is there any hope of estimating the severity of reinfection based on evolutionary data?
Yeah, I'm glad you asked that question. I thought that question while giving this talk to you. I didn't think of it before, though, but while giving this talk, I was thinking, oh, yeah, actually, maybe. So our minds are in the same place, whoever put that in the chat. I think there is some hope of that. That's a little trickier. It involves, it's a little trickier just because we don't need to just come up with a durability of immunity. We need to sort of really understand the immunological component of your level of antibody and how that affects how sick you get, which is just, I think, a much more nuanced question compared to how long you remain immune. So, but in principle, there's no reason why we couldn't do that. If we can sort of associate the level of antibody to the amount you get sick, then we could do that on this, and in this approach, then we could do that using this approach seamlessly. I think it's something that's perhaps challenging to do. Maybe people just haven't really tried to do it. I'm not sure. Again, I really want to lean on my immunologist collaborators to understand that a little bit better.
Thank you, Jeff, so much for a wonderful talk, and glad everybody could come today. Just a reminder that in 2 weeks, on September 24th, we'll have our next one that will be from Ramanan Laxminarayan at CDDEP. So hope to see everybody there.
I really enjoyed your questions, everyone. Thanks for listening to the talk.
Thank you so much, Jeff, for doing this. We appreciate it.